Now, after transcription is done, in prokaryotes, remember that transcript is ready for translation, no problem. There, ribosome can hop on and start making the polypeptide. In eukaryotes, it's not the case, right? In eukaryotes, transcription makes this pre-mRNA, or this primary transcript that needs to be modified before it can be used for translation. So let's take a look at what kind of processing this RNA needs to go through before it can enter the cytoplasm and be used for translation. This RNA processing comes in two parts, and we'll talk about both of them in more detail in the next few slides. Part 1 is modification of both ends of the mRNA. The 5' end is going to receive what is called a 5' cap, and then the 3' end, where that polyadenylation signal sequence was, it gets more adenines added to it. It's called the poly A tail. And then number 2, the RNA is going to have to go through a process that's called RNA splicing. So in eukaryotic genes, you don't have one solid gene that codes for protein. You've got a section of the DNA with chunks that code for protein with non-coding pieces interspersed in between. And so these non-coding sequences in the DNA are called introns, and the coding sequences that do code for protein are called exons. I like to think of it as exons, EX. Those are the parts of the RNA that actually exit the nucleus and are translated. And the introns, INT, are kind of intervening. They're getting in the way of the coding parts of the gene. And so what the cell needs to do is it needs to remove all the introns and stitch together or splice together all of the exons to make a one continuous mRNA that actually, the whole thing codes for amino acids. So let's take a closer look at modification of the ends of the RNA first. So down here we've got our primary transcript. In orange, you'll notice on the 5' end, the 5' end will receive a 5' cap. This is a modified guanine nucleotide, and it's attached actually with all 3 of its phosphates. And then on the opposite end, the 3' end, you'll see here's the polyadenylation signal sequence, and past that, the mRNA will receive a poly A tail. This is anywhere from 50 to 250 extra adenine nucleotides added to the 3' end. And both of these end modifications seem to play really important roles for the mRNA in the cell. First of all, they seem to help the export of the mRNA out of the nucleus into the cytoplasm. Also, remember that single-stranded nucleic acids are highly subjected to degradation, and so these end modifications seem to protect the mRNA from these hydrolytic enzymes, so the RNA isn't degraded as soon as it's created and can actually survive until it can be translated. And then lastly, both of the end modifications can help a ribosome attach to the 5' end to initiate translation. Now let's talk about RNA splicing. So here we've got marked the exons and the introns. So the exons, or the coding regions, are in dark orange, here, here, and here. And then the introns, the intervening sequences that don't code for amino acids, are shown in the light orange. And so what the cell needs to do is it needs to cut out this intron, and then stitch together just the exons, or the dark orange coding parts of the mRNA. And so this then will complete a fully functioning and mature mRNA with a 5' cap, poly A tail, and only the coding parts of that gene stitched together. So only the exons remain. Now, what actually carries out the splicing that takes place for the mRNA in cells is a very large structure called a spliceosome. The spliceosome is interesting because it is made out of 2 different kinds of macromolecules. So it's part protein, shown here in purple, and also part RNA, which is shown in orange. And in this case, in the spliceosome, it is the RNAs that are the catalytic part of this structure that can actually catalyze the reactions of cutting out the introns and stitching together the exons. And so we give these catalytic RNAs a very special name. We call them ribozymes because they are catalytic RNAs, whereas typically you think of most enzymes as being proteins. And so the spliceosome, its main function is to take an mRNA primary transcript, figure out where the intron and exon junctures are, cut out the introns, and then stitch together the exons to make sure that you have a solid mRNA that only has the coding regions in there. All right, and so hopefully, I'm hoping somebody's thinking, well, gosh, that seems wasteful, right? It's going to require energy on the part of the cell to actually cut out all these introns. What's the point in having them in there anyway? Why not just have one complete gene that just holds the coding regions of a particular protein? And this is because eukaryotic cells, many of them, can go through a process that's called alternative RNA splicing. Humans, our cells, they can do this. This is what allows us to get away with less DNA than an algal cell and be able to do things like talk and drive a car and do calculus. So what alternative RNA splicing is choosing which exons to keep at different points in a cell's life, right? So you can take one gene and you can make multiple different proteins out of that same gene. So you don't have to carry a lot of genetic information. You have a little bit of flexibility in the genetic information that you carry, meaning that you can create different products from that same gene. So if we were to take a look at this, right, so in this example, we've got a gene here and it's coded, it's got 1, 2, 3 exons. And typically each exon for a gene will code for a different domain of that particular protein. Right, so here exon 1 codes for domain 1, exon 2 codes for domain 2, and exon 3 codes for domain 3. Let's say the cell says, well, I don't need all 3 domains today. Based on the environment that I'm existing in currently, I only need domains 1 and 2. What the cell can do is it can go through alternative splicing, and it can, instead of keeping exon 1, 2, and 3 when it splices its RNA, it can just keep exon 1 and 2 and get rid of exon 3 as if it were an intron. Now you create a slightly different mRNA. You also create a slightly different protein that's missing that third domain that has a different function within the cell. So again, alternative splicing gives cells the ability to create different versions of proteins from just that one single gene by picking and choosing which exons that cell actually wants to keep for translation.